Hello and welcome to the GenQA Focus On webinar, where we're going to present some data and information around RET testing. This webinar is supported by Eli Lilly. First of all, I would like to welcome our guest speaker, Dr. Jaclyn Hechtman from the Memorial Sloan Kettering Cancer Center in the United States. And Jaclyn has very kindly prepared some background information and a presentation on RET testing. Also, please welcome my colleague, Dr. Jenni Fairley, who's the Deputy Director of GenQA and runs and manages the Molecular Pathology External Quality Assessment Schemes. And very kindly, both Jaclyn and Jenni have agreed to participate in a live question and answer session at the end of the presentations. So please do add any questions that you would like to be raised into the chat box or into the questions box that you can see hopefully on your screen, and we'll be able to spend some time and get some feedback from our presenters then. Please can everyone go on mute and turn your cameras off, just because we have many people on the call and it just makes life a lot easier. And obviously don't put hands up or flag for questions, because again, as I say, we'll deal with all the questions together at the end of the presentations. So we're going to start off with an introduction to RET, testing for RET fusions, and then a comparison of the different RET testing methods, because that's always very interesting to determine the limitations of different approaches for testing. And then Jenni is going to cover the results from previous external quality assessments where RET testing has been assessed, and then we'll have our live Q&A session. So welcome again. I'm Professor Sandy Deans. I'm the Director of GenQA, and I have great pleasure in now introducing Dr. Hechtman to give her presentation. Thank you.
Hello, and thank you for tuning into this webinar. Today we are going to review targetable alterations in the RET gene and the assays used to detect them. Before we start, I'd like to introduce myself. I am Jackie Hechtman, and I do molecular and surgical pathology at Memorial Sloan Kettering Cancer Center in New York City. My Twitter handle is Jackie Hechtman, or you can find me by scanning this QR code with your smartphone camera. So the information presented in this webinar is being presented at a personal capacity and does not represent the opinions of my employer. Relevant to the current webinar subject, I have received speaking honoraria from Illumina and Eli Lilly, as well as a research grant from Eli Lilly. I have also received unrelated honoraria and research funding listed here. The objectives of this activity include: 1, describing the evolving landscape of biomarkers in non-small cell lung cancer, with highlight on oncogenic RET alterations. 2, being able to list the prevalence of RET fusions and hotspot mutations across different cancer types. 3, understanding the response rates and molecular eligibility criteria for approved RET inhibitors, and most importantly, 4, listing the strengths and pitfalls of different assays clinically used to detect RET fusions, including FISH, IHC, DNA-based NGS, RNA-based NGS, and combined DNA-RNA-based NGS. So non-small cell lung cancer, which I will refer to as NSCLC from here on out, is mainly composed of adenocarcinoma and squamous cell carcinoma, while about 20% of cases are other histologic types. While squamous cell carcinomas only rarely have mitogen-activated protein kinase pathway drivers, the majority of adenocarcinomas have these MAPK pathway driver alterations. They include KRAS and EGFR mutations most commonly, while kinase fusions occur in just under 10% of lung adenocarcinomas, and RET fusions within that group occur in 1 to 3% of lung adenocarcinomas. These results have shaped current molecular testing recommendations for NSCLC. Currently, all patients with lung adenocarcinoma and younger and/or non-smoking patients with squamous cell carcinoma should receive testing for EGFR mutations, ALK and ROS1 fusions, BRAF mutations, and as well as PD-L1 IHC for TPS scores. Broad panels are recommended over single gene testing, as biopsies often are limited in terms of the amount of tumor tissue present. The 2018 recommendations list KRAS, RET, HER2, and MET testing as appropriate as part of larger panels, but this is likely to change with current therapies for patients harboring KRAS, G12C mutations, MET exon 14 skipping mutations, and RET fusions showing really promising results. So the RET gene is located on chromosome 10 and stands for rearranged during transfection, as the DNA sequence of this gene was originally found to be rearranged within a 3T3 fibroblast cell line following its transfection with DNA taken from human lymphoma cells. This gene encodes a receptor tyrosine kinase that is physiologically important in neural signaling as well as kidney development. While loss of function mutations are associated with Hirschsprung disease characterized by the absence of ganglion cells in the colon in babies, activating alterations are present in cancer. Activating mutations are present in the germline of patients with multiple endocrine neoplasia types 2A and 2B, which are characterized by pheochromocytomas, medullary thyroid cancer, and parathyroid gland neoplasia. Activating mutations can also be sporadic in medullary thyroid carcinoma. Sporadic RET fusions have been identified in many different types of cancer, but most notably present in non-small cell lung cancer. Under physiologic conditions, extracellular ligands of various types activate the RET protein in a regulated manner. However, activating RET missense mutations and fusions joining the kinase domain of RET to a different gene result in constitutive ligand-independent signaling with downstream activation of the PI3K and ERK pathways, with resulting uncontrolled cellular proliferation. Structurally, the vast majority of RET protein fusions occur via intrachromosomal inversions that place the 3' segment of RET, including the kinase domain, distal to another gene, most commonly KIF5B in lung and NCOA4 in CCD6 in papillary thyroid cancers. The reading frame is preserved between the two genes, and the fusion results in constitutive signaling and MAPK pathway activation. These illustrations demonstrate which exons are involved in various common RET fusions. When KIF5B is a 5' partner, as often seen in lung cancer, RET exons 11 and 12 are often the first exon of RET involved, which means that the breakpoints may occur in introns 10 and 11 of RET, respectively. For the other partner genes, so exon 12 is the most commonly RET exon involved, which means the breakpoint in RET intron 11 is most likely. That means that a DNA-based NGS test assessing RET fusions needs to cover RET introns 10 and 11. Inherited and sporadic activating RET mutations are found in the majority of medullary thyroid cancers. The most common inherited mutation occurs at codon C634 in the extracellular portion of RET. Mutations occurring at codon M918 in the kinase domain are the most frequent mutation in sporadic medullary thyroid cancer, but have been found in MEN2B patients as well. Mutations in either codon have been correlated with aggressive medullary thyroid cancer. Several other mutations throughout RET have been identified as activating in patients with hereditary endocrine malignancies. So this bar graph emphasizes the frequency of missense mutations in medullary thyroid cancers, and you can see that the majority of cases have it, or at around 80%, as well as the frequency of fusions, which are listed here in purple, in different types of non-small cell lung cancer, with lung carcinosarcomas harboring RET fusions at a rate around 15% and adenocarcinomas at a rate less than 5%. Recently, a group at MSKCC in New York took a deeper look at RET fusions at our institution. We identified a total of 151 RET fusions. The majority of these fusions occurred in NSCLC, while thyroid cancers, mainly papillary, had the second highest incidence or prevalence of RET fusions. The three major partners that have been previously discussed accounted for approximately 87% of RET fusions, with KIF5B accounting for 45% of fusions, CCD6 fusions accounting for about 29% of fusions, and NCOA4 accounting for about 13% of fusions. More specifically, KIF5B only occurred in lung cancer, accounting for about 69% of RET fusions in lung cancers. CCDC6 accounted for about 63% of RET fusions in thyroid carcinomas. Interestingly, the time of diagnosis is not the only time that RET fusions need to be screened for. Recently, fusions have been identified as a mechanism of acquired resistance to EGFR inhibitors in lung cancer. While these have only been recently discovered and treatment rates are low, patients who have received targeted therapy addressing the new fusion demonstrated clinical benefit in 2 out of 2 cases listed here. So now let's shift gears for a few minutes and talk about the selective RET inhibitors currently in use. In 2018, the results of LOXO-292, also known as selpercatinib, were released. This small molecule inhibitor had an objective response rate of about 60% in lung cancer patients with RET fusions with various partners. The responses were durable, with a median duration of response not reached by the time of presentation of this data. Blue 667, also known as pralsetinib, was also presented in 2018 with very similar results to LOXO-292 for RET fusion positive lung cancer patients, as shown here. As mentioned previously, LOXO-292 showed durable responses, with some patients still on treatment, as can be seen from the swimmer's plot, for over a year at time of result presentation. Outside of NSCLC, thyroid cancers also demonstrated promising results for both RET fusions in papillary thyroid and RET mutations in medullary thyroid cancers. And similar to non-small cell lung cancer, the responses were again durable for both fusions and mutations in thyroid cancer, as can be seen here, with the fusions in purple and the mutations in blue. Other cancer types with RET fusions at very low frequencies, such as pancreatic cancer, cholangiocarcinoma, and infantile fibrosarcomas, have also demonstrated clinical benefit from these 2 RET inhibitors. Selpercatinib, as we said, also known as LOXO-292, and Retevmo received FDA approval for patients with metastatic RET fusion positive lung cancer, RET mutant medullary thyroid cancers requiring systemic therapy, and RET fusion positive radioactive iodine refractory thyroid cancers requiring systemic therapy. Pralsetinib, also known as Gavreto, was approved just 3 months later. The indications for those are very similar to selpercatinib. So now that we understand why RET detection is important, we'll spend the remainder of this activity discussing clinical assays currently in use to detect RET fusions. This slide shows the most commonly used assays in practice today to detect RET fusions. We will discuss the sensitivity and specificity of these assays later during this talk. Fluorescence in situ hybridization, or FISH, usually employs break-apart probes and only requires 1 slide. Because it is DNA-based, it can work on tissue samples that are older. Turnaround time is often 2 to 3 days. IHC is the fastest and cheapest of all the methods. FISH and IHC both use visualization, which means that low tumor purity is okay for these 2 assays. For reverse transcription PCR, along with the other RNA-based methods which utilize the extraction of RNA with subsequent creation of a DNA library, mean that due to the nature of RNA, blocks should generally not be more than a couple years old because RNA is a very labile nucleic acid. So RT-PCR is specific, yet sensitivity depends on whether the partner gene is covered by the 5' partner. So the partner gene as well as the breakpoint exon need to be tiled in. DNA-based NGS has the advantage of simultaneously giving results for other mutations, tumor mutation burden, sometimes microsatellite instability status, amplifications, and other structural variants. The downside is that it is more expensive than real-time or reverse transcription PCR and generally takes 1 to 3 weeks for results, with amplicon-based NGS being faster and cheaper, but better for hotspot mutations and other genetic aberrations in comparison to hybrid capture-based NGS assays. There's also anchored multiplex PCR, which is an RNA-based test, and unlike reverse transcription PCR, it works agnostic of the 5' partner, thus increasing diagnostic sensitivity. Lastly, there is combined DNA RNA-based NGS, which is the newest of this group of tests. In our institutional study, we examined the sensitivity and specificity of 3 clinical assays: DNA-based NGS, break-apart FISH, and IHC for the detection of RET fusions. The results were compared against our RNA-based NGS assay, which is based on anchored multiplex PCR technology. We found the sensitivity of the DNA hybridization-based capture assay was 100% because it was possible to target all exons and introns involved at breakpoints. Specificity was very high, yet there were several RET rearrangements detected by DNA-based NGS that didn't result in fusions at the RNA level. These DNA level rearrangements often did not include the kinase domain of RET, which were predicted to be out of frame and involve novel partners. RET fish had an overall sensitivity of 92%, with 100% sensitivity for all fusions except those involving NCOA4. NCOA4 RET fusions were only detected around 2/3 of the time, and specificity was 72% in this cohort for fish. And the reason behind the low specificity of fish in the study was that we looked at the RET rearrangements detected by DNA-based NGS that didn't result in fusions by fish as well. And indeed, fish did confirm that there were rearrangements in these cases. IHC performance was somewhat similar to fish, with 100% sensitivity for KIF5B fusions, but only around 50% sensitivity for NCOA4 fusions. And it was approximately 89% sensitive for other fusion partners. Specificity was tested in NSCLC, papillary thyroid, and medullary thyroid carcinoma, and specificity was approximately 80% in NSCLC and papillary thyroid, but it was completely nonspecific in medullary thyroid cancer. So now we'll take a deeper look at each assay. Break apart FISH is a DNA-based assay that involves visualizing two different colored probes that flank the gene of interest, in this case, RET. If the RET gene is intact, the two probes, one red and one green, look yellowish in each tumor cell, and you should have one for each copy. However, if RET is rearranged, there is one red probe and one green probe visually. Technically, only one slide is needed, and only about 100 tumor cells need to be visualized regardless of purity. Because each cell is visualized, low tumor purity is fine. Turnaround time is relatively quick. However, break apart FISH detects rearrangements and is not specific for oncogenic fusions, as it only tells you if RET is not where it is supposed to be. So the potential problem with the sensitivity of RET FISH lies in the distance of the fusion partner to that of RET. All three of the most common partners in the yellow boxes here are on the same chromosome as RET, which is chromosome 10. But as you can see here, NCOA4 is very close, and only approximately 2/3 of these fusions can be detected by FISH due to the subtlety of the split signal. In this slide, one can see a clear rearrangement in the top panel as the red and green probes are split very far apart, and this is an example of a KIF5B RET fusion. In the bottom panel, the red and green probes have a very subtle split pattern and are very close together. This was an NCOA4 RET fusion, and this case did not meet clinical criteria for rearrangement calling. RET IHC is not as commonly used as a screen for RET fusions at the current moment, and studies on it have been varying with respect to sensitivity and specificity. And prior to now, most studies were pretty small. For RET IHC, the epitope must be in exon 12 or distal because exons 1 through 11 of RET are generally not part of the fusion protein. The antibody generally should be monoclonal to reduce interbatch variability. So we investigated EPR2871 as a potential clinically useful RET IHC antibody. And we found that at the definition of positive cytoplasmic staining, IHC is very sensitive for KIF5B, around 90% sensitive for CDC6 RET fusions, and the least sensitive for NCOA4 RET fusions at 71%. The specificity generally was around 80%. We found it not to be useful for medullary thyroid because it stained every case regardless of mutation status, and that might be due to the neural differentiation. Because it's visual like FISH, it'll work for low tumor purity cases. The images here are some examples of how RET IHC stained. KIF5B RET fusions were often showed very strong cytoplasmic RET staining and expression, while NCOA4 and CCD6 RET fusions were weaker. If only macrophages or luminal only staining patterns were present, then we did not count these as positive. They were counted as negative because we found these to be very nonspecific. This is an example of reverse transcription PCR, in which RNA is extracted and converted to a cDNA library. Good quality RNA is needed along with an internal control, and several slides are needed for extraction unless nested RT-PCR is performed. And it's very possible here to detect RET fusions even if tumor purity is very low. So that's another good aspect of this particular assay. So with that, DNA-based NGS is actually okay for RET fusions because introns 10 and 11 of RET are possible to cover. Because tumor is scraped from a slide rather than visualized, low tumor purity can be a significant issue, and multiple unstained slides are required depending on the specific assay. Some of the advantages include the fact that other oncogene mutations, other fusions, tumor mutation burden, some amplifications, and microsatellite instability status can often be simultaneously assessed through DNA-based NGS. One downfall is that sometimes a RET rearrangement might be identified that needs further testing because the kinase domain is not included, or the partner is novel, and/or the fusion is not predicted to be in frame. When this happens, a backup assay is needed, and probably the best backup assay would be an RNA assay that looks specifically at whether this rearrangement results in a transcribed fusion. This slide demonstrates some differences between amplicon and hybridization capture-based NGS. While hybridization-based capture takes longer and is more expensive, larger regions of interest can be looked at, and less PCR steps make intron tiling for fusions possible and copy number easier to analyze. NGS can be performed with DNA or RNA as a starting point. Because fusions most often have breakpoints that occur within the introns, DNA-based NGS needs to cover all the introns that might be commonly involved. Introns can be long and repetitive with some genes like NTRK3. However, for RET, we don't really have that problem as much. When RNA is extracted, the introns are not present in the cDNA and the exons are assessed only, which makes the testing somewhat simpler. And this slide summarizes some of the issues that cause DNA-based NGS to be less sensitive for fusions than RNA-based NGS. Issues, as we said before, like large and repetitive introns for some genes like NTRK3, lower tumor purity, and complex events make DNA analysis more difficult. With this knowledge in mind, we assess the performance of our in-house DNA-based hybridization capture-based assay for RET fusions. And we found that 12 out of 32 structural variants of uncertain significance did result in fusions on further testing. Factors including high read count, a breakpoint in intron 11, and the lack of other MAPK pathway driver alterations all made the likelihood of uncovering an oncogenic RET fusion more likely. So in 2014, anchored multiplex PCR was published as a novel assay for looking at fusions with RNA. This assay assists RNA with a gene-specific primer for the kinase gene and exon, and a universal primer that allows novel fusions to be discovered. Because fusions are often accompanied by upregulation of the mRNA levels, fusions can often be identified even in the context of lower tumor purity. Aside from separate DNA and RNA-based NGS assays, there are several commercially available combined DNA, RNA, NGS assays, and this slide lists and describes several of the ones currently available. I also want to mention that the European Society of Medical Oncology recently put forth guidelines for RET fusion testing. NGS is recommended when available, with FISH or RT-PCR as a backup method. The guidelines can be found in the journal Annals of Oncology. Our paper came out at a similar time, and unfortunately, our two papers didn't really overlap. Fortunately, the RET fusion algorithm we proposed is similar to the one proposed by ESMO. However, ours includes some added reflex testing to account for the pitfalls that we've seen for each assay. So, for example, for DNA-based NGS, if the structural variant is of uncertain significance, then further testing might be useful. And the same thing for a FISH screen with a negative but subtle split pattern. ESMO also put forth RET mutation testing guidelines, and they can be found in the journal Annals of Oncology as well in the same paper. So to conclude, oncogenic RET fusions and mutations are important to detect because 2 FDA-approved inhibitors with high and durable responses are now available. These fusions are most commonly seen in papillary or poorly differentiated thyroid cancer, as well as lung adenocarcinoma, with mutations being seen in medullary thyroid cancer. But fusions also occur rarely across other cancer types. There are various assays available for the detection of RET fusions, and the advantages and pitfalls of these assays were summarized in this webinar and in previous slides that will be available online. Because each assay has different advantages and pitfalls, it might be best to consider having 2 validated assays available in labs with high volume. The first choice assay depends on the amount and quality of tissue available, other biomarker assessments needed, the type of tumor, and how soon a result is needed. And with that, this presentation is finished. Thanks so much for listening.
Thank you, Jaclyn, very much indeed for such a well put together presentation that just gives an idea of why we do RET testing and how we should be doing RET testing. So thank you very much indeed. So again, if anyone would like to ask some questions, there's some now coming through from you, please pop them in the questions box and we will cover them in the live Q&A session at the end of the presentations. So it's now my great pleasure to introduce my colleague, Dr. Jenni Fairley, who is going to give a summary of the findings from the external quality assessment schemes run for RET testing. So Jenni, over to you now, please. Thank you.
Thanks, Sandy. As Sandy says, I'm going to summarise the findings of our previous RET EQAs. So we first started running EQAs, which included testing of RET in 2015. We set up a new pilot EQA for looking at some of the novel biomarkers which were of interest in lung cancer. So in that EQA, we included ROS1, RET and MET testing. And we had enough interest in this EQA to keep running it for a few years afterwards, as you could see. So in 2016, we actually ran another 2 rounds of this EQA. In 2017, we had run enough runs of the EQA to make us confident that it was fit for purpose, and therefore we could introduce it as a full EQA. And then in 2018, we went on to do one more additional round of this as a full EQA, and again in 2019. And then in 2020, we actually undertook a reorganisation of some of our EQAs for lung cancer. And I'm just going to summarise what we saw with those. So in 2020, we merged together our additional lung biomarkers EQA with our other molecular biomarkers lung cancer EQA to form basically a suite of lung cancer EQAs, which labs could choose the one that was most suitable for their clinical needs. So there are still a number of laboratories who would only test for EGFR, who were only testing for EGFR, ALK and ROS1, because those were the biomarkers which were licensed for use in most countries at that particular time. We offered an EQA that only looked at fusions, so for ALK, ROS, RET, and then also MET amplification or exon 14 skipping. And then we offer a comprehensive EQA, which we looked at the most clinically relevant biomarkers for lung cancer testing, and that's part of them. So RET testing in 2020 was included in our additional lung biomarkers and in our lung cancer comprehensive, so our fusions and our comprehensive EQAs. And over the years, we've seen a big increase in the number of participants who actually did RET testing in these EQAs. So we started off with a very small number. There were only about 5 labs who reported results for RET in that first run of the EQA we did in 2015. And this increased, and there were actually 49 labs that reported RET testing in 2020. And you can see there's been quite a dramatic increase when we introduced RET testing as part of our lung cancer EQA between 2019 and 2020. And that's also in a large part due to the testing performed by the laboratories, which is summarised in the following slide. So in our first runs of these EQAs, the vast majority of laboratories were performing FISH testing to look at RET. So that's in the left-hand side of this graph here. Again, there were small numbers of laboratories using each different type of FISH probe, but it was the most popular. And we only had a handful of labs performing NGS up until really last year in 2020, we've seen a dramatic increase in the number of labs doing NGS. That's not surprising that now there's more biomarkers out there that require next generation sequencing testing, and that's reflected in what we see with this. And in fact, about 75% of labs doing testing for RET in our 2020 EQA use next generation sequencing. It's much smaller numbers now doing FISH. So the results of the EQA were as followed. So in 2015, we sent out 2 cases for RET testing. One case did have a RET rearrangement on a fusion transcript, and the other didn't. And all the laboratories reported the correct results. As I said, in 2016, we had 2 EQA runs. In round 1, we sent out 3 cases, and none of them had any fusions or rearrangements. And again, we didn't see any critical errors. In run 2, we actually sent out 1 case that had a fusion and a rearrangement, and the other 2 didn't. But one of the 4 laboratories that used FISH for this EQA didn't report the rearrangement in this case. And I'll probably come back to that. In 2017, we didn't send out any cases with a rearrangement, and there were no errors. But in 2018, we sent out 3 cases. And 1 case had a rearrangement, a fusion, and the other 2 didn't. But 3 out of the 8 labs who reported FISH results didn't report the rearrangement. And similarly, in 2019, when we sent out 2 cases, we had 1 case that had a fusion and the other one that didn't. And 3 out of the 12 laboratories performing FISH didn't report the rearrangement. And then in 2020, we sent out 3 cases. None of those had a rearrangement or fusion, but and again, we didn't see any critical errors. So I think the positive note that nobody was reporting any false positives in any of these EQAs. However, as I mentioned, we did have a few labs performing FISH, which didn't report the presence of the fusions. So in particular, for the 2018 and 29 EQAs, these EQAs, we sent out a sample which had a CCD6 RET fusion. The breakpoints of this fusion had been identified and were sequenced, and we had the samples validated using FISH and NGS, and the rearrangement was shown to be there using the FISH that our validating laboratories used, and the NGS confirmed the presence of the fusion. However, it seemed that the detection of this rearrangement was FISH probe dependent, because it was particular FISH probes were unable to detect the fusion there. However, when we went on to do a little bit further digging into why this might be the case, the samples we used were from artificial cell lines, and we couldn't rule out that because these could have been made in a way that would cause an artifact in them that would cause this discrepancy in the FISH results. We therefore couldn't give the laboratories poor performance for failing to report these rearrangements, and we don't have any evidence that actually there's any problem with any of these probes. We think it could be an issue with the actual material, which just demonstrates the issues that we can see when using artificial material. So in general, throughout these EQAs, we have noticed some common issues with reporting of fusions, and particularly related to NGS, some of these. So laboratories will often use genomic coordinates to report results. But if they do that, they've got to remember that you have to include the genome build, otherwise the genomic coordinates are going to be incorrect or are not going to mean anything. A recent Hugo guidelines have recommended that fusions should be described using a slash between the two genes rather than a dash, as might have been the case in the past. So a CCDC6/RET fusion. If laboratories report the breakpoints of fusions, i.e. nucleotide positions or exon numbering, the reference sequences for the genes used should be included in the report. And if appropriate, HGVS or ISCN nomenclature should be used. We haven't assessed any of our EQAs on the basis of HGVS nomenclature for fusions yet. The latest version of HGVS, which was released in May last year, does include nomenclature to describe RNA fusions using the double colon to indicate the fusion, with numbering according to the coding sequence of the transcript. And the number 1 position being the A of the ATG codon, which is really important to be aware of. It's not the transcription start, so it's not what the transcript starts with, but it's the 1 of the ATG, as is true for other types of HGVS nomenclature. If only the breakpoints are sequenced and not the entire transcript, a question mark can be used to describe the start-end positions. We're not going to mark fusion nomenclature yet in our EQAs, and we're not going to expect laboratories to compulsorily include this, because we appreciate it's quite new and it's going to take a while to get used to, but we'll definitely be monitoring that for the future. So in 2021, once again, we'll be running our lung cancer EQAs for the comprehensive EQA and the fusion EQA, which both include testing for RET rearrangements or fusions, and it's suitable for using FISH, NGS or RNA-based methods. And for 2021, we'll have 2 EQA runs with 5 samples in round 1 and 4 samples in round 2. In 2020, we did decrease the number of samples we sent out due to COVID-19. And then we have a new pilot EQA in 2021 for thyroid cancer. And that will look for RET fusions and rearrangements, again using NGS and RNA-based methods, but also for RET SNVs as well. So as described for medullary thyroid cancer. Online enrolment for these EQAs is closed now, but if you're interested in participating in them, please contact us and we may be able to accept your participation into the EQAs. And it's just reminds to thank our specialist advisory group who help us determine how we should be delivering EQAs, and also our assessors, who we couldn't really do this without, because it's them that decides the scoring for all the EQAs, and our samples sourcing and validation laboratories. Again, we couldn't do it without them, because we're very reliant on them for providing us with samples to use for the EQAs and the like. I want to thank Lily for supporting this webinar, and most of all our participants, because they're a very important part of who our EQAs are for. And now if you've got any questions, there's still a chance for you to ask them in the questions box. And I'll pass you back to Sandy.
Great, thank you, Jenny. That was a really nice summary and a whiz round the RET EQAs that we've been providing. Thank you for that. Really interesting to see the shift in methodology as well. So we've got some questions coming through. So let's have a start. So Jaclyn, I've got a couple for you. So you mentioned about the testing approach really should be based on how quickly you need the result, the clinical application, how much materials available, etc. So would you recommend IHC to be a quick and dirty first screen for RET fusions and mutations, as it's so quick and cheap and easy to do?
Thanks for that question. Currently, IHC is not recommended. So there's only been limited data on it. And because the sensitivity is not very good for NCOA4, although it is very good for KIF5B, you might be missing certain targetable fusions. And in particular for mutations, the specificity in medullary thyroid cancer seems to be not good at all. So IHC isn't really recommended. If time is of the essence and material is limited, FISH might be a better idea, or RT-PCR if it's available at your lab.
Sure. Great, thank you for that. That makes a lot of sense. But surely IHC must have a role, or potentially have a role, in looking at whether or not the fusion would be translated or not, looking at the end product of the protein. Is that something that you have heard happening?
Right, so I think it can be helpful there. Sometimes you have a structural variant of unknown significance in the DNA and you don't know if it's going to be transcribed. So ways to look at it include looking at RNA, which would be the ideal methodology. So if you have something in the DNA where you have a variant, you need to look at either the RNA or the protein. And generally, the specificity is about 90%. So IHC can indeed help when you have a structural variant of unknown significance in the DNA. However, RNA is probably the most ideal if you can do RNA-based NGS or RT-PCR involving all the exons of RET in the partners.
Sure. Okay, thank you for that. A couple more questions for you, Jaclyn. We'll stick with you. So Pauline, hi Pauline. Is it possible to increase the amount of tissue sent for testing? Many of the NGS panels require significant amounts of DNA and RNA that is not possible to extract, so we sometimes revert back to FISH. Is that something you experience in your clinic?
Is it possible to increase the amount of, oh, do you mean how to get more tissue?
Yeah, basically, yeah. And I think this is a recurring theme. Somebody else has put in the chat, for NGS, it's a lot. So how many biopsies don't actually meet your criteria for NGS testing?
It depends on the quality also. So even if the sample's small, I think, if the sample is good quality, the vast majority, perhaps like 90%, if you have 20 unstained slides, should be able to make it. So it depends on a few things, the quality of the material as well as the tumor purity on a DNA-based test. So those things are both important. And on a biopsy, I think the main way to increase the amount of tissue you're getting is to scrape all the tissue if purity is not an issue, and have more slides sent. So rather than like 5 or 10, if it's a very small biopsy, try to do 20, 25. Sure. You can also try not to trim the block. So if you're doing IHC and molecular, you can have all the unstained slides cut at once. So you're doing PDL1, for example. So you cut this unstained slide for that, as well as unstained slides for molecular at the same time to not cut into the block further.
Yeah, so you're not wasting sections. Yeah.
Yeah, so that might help.
Great. Paulina has come back again saying she also meant for EQA purposes. So Jenny, where are we about supplying more material for NGS testing labs for this EQA?
When we send out slides for labs who test both DNA and RNA, we will provide them with some more sections now, because we appreciate if you're getting DNA and RNA, you do need a bit more. However, all our sections have been validated by laboratories using appropriate methods with the amount of material we send out. And, for example, in that last EQA data I provided you with, where there were 49 labs doing RET testing using NGS, they were all using the amount of material we sent out and were able to get results. So there's always a balance with this as well. And we're trying to reflect a clinical picture where if you're testing small biopsies, actually the material we provide tends to be more tumor rich and have a bit more cellular than you would see with the biopsy as well.
Okay, thank you, Jenni. Also, another question for you, Jenni, about EQA. So what does GenQA, the EQA provider, actually do if you pick up an error? So a lab has reported the incorrect results. I know none of them were acted upon in the previous runs, as you explained, but what does actually happen? Do you do nothing? Do you actually follow it up? What's the process?
So in the first instance, if we see an error within an EQA, and say we saw quite a few labs that were missing the RET fusion, we would actually investigate that internally to start with anyway. We know which errors are made in our EQA, and we're always very careful when we're using real tissue that that's not due to some kind of heterogeneity, so we will always do internal investigations. The laboratories have the opportunity to appeal if they think we've made a mistake. And then if that's confirmed that the laboratory has made an error, we will run that by our specialist advisory group to confirm the error. And then we follow this up with a root cause analysis. So we ask the laboratories to investigate their root cause analysis. And if they need any assistance with that, we're quite happy to provide any laboratories with assistance to investigate any problems they have in their testing. For more serious errors, where laboratories will make errors in more than one EQA run for the same type of testing, we can go on to further refer the laboratories, certainly within the United Kingdom, to NECWAP, committee of the Royal College of Pathologists, who will investigate further their poor performance. But we're always very happy to help laboratories investigate any issues they've had with EQA performance by contacting us.
Okay, thank you, Jenni. That's good to know that we're there to help them as well. Jaclyn, a couple of questions for you. So this is more about reporting a structural variant. What sort of parameters or limitations do we need to make sure are on the report when you've identified a fusion, either by DNA or RNA-based NGS?
Sure, it's particularly important for DNA-based structural variants to have specific parameters. So you want to say whether it's predicted to be in frame or out of frame, and whether or not the kinase domain of RET is included, and what the partner is. So you can basically say whether or not, you can also say whether this is novel or has been described in the literature before as an oncogenic fusion, or whether or not its oncologic significance is unknown, and then also whether you need to recommend doing reflex testing with RNA-based methods.
Great, thank you. And I guess, Jenni, you see different approaches and content on reports through the EQA scheme as well from the many labs that participate?
That's right. I mean, one of the things we always think is quite important, if you don't find anything to make it clear what testing you've actually done, sometimes it's not always very clear that there might be other reasons why you haven't picked up something in your sample. And again, the tumor content of the sample is really critical for that. To know you might not be enough tumor cells might be the reason why you're not picking up a fusion in your sample. We see quite a lot of laboratories that don't always include that in the reports, but it's a really critical piece of information.
Yeah, thank you, Jenni. And I guess we see that right across the board for the molecular pathology testing schemes as well. It's not just the RET testing.
Indeed.
Thank you. So, Jaclyn, one final question to you. So you mentioned that obviously there's a high percentage in the hereditary and sporadic melanoma thyroid cancers, and non-small cell lung cancer RET testing is now coming into mainstream. What are there other types of cancers that should be tested for RET fusions and RET mutations?
So at the current time, at least in the US, the types of cancers approved for RET inhibitors would include lung cancer, medullary thyroid cancer, and papillary thyroid cancers, so papillary thyroid cancers and lung cancers have RET fusions, and medullary often has these mutations, and there's also other types of thyroid cancer that could be treated. While other types are not yet approved for targeted therapy, we sometimes do see RET fusions in certain types of cancers. So for example, we see kinase fusions enriched in MLH1 deficient colon cancers that are BRAF wild type, and if the patient is negative for Lynch. So once those are all mutant, they're highly enriched for kinase fusions, including RET. And then we've seen several breast cancers that have had kinase fusions, including RET fusion after progression on endocrine therapy.
Great, thank you. And I guess as the RNA fusion panels become more mainstream, then there's a higher chance of picking up different fusions, including RET fusions in other cancer types that are happening to go through the panel as a high throughput service.
That's right. You'll even see them on the DNA-based NGS assays that are longer and tile in the RET kinase domains, including the introns and exons. And then, of course, you'd see it also on the RNA-based assays.
Yeah, great. So much information to be gained. That's great. Thank you very much. Thank you, Jaclyn and Jenni, for a fantastic session. I've definitely learned a huge amount, and also for giving your time to do this live Q&A. I think it's a really special part of the webinar to be able to respond to the live questions as they're coming in. So thank you both very much indeed. And Jenni, I'll hand back to you just to close the session, please.
Thanks very much, Sandy. Well, because you've logged in using GoToWebinar, you should receive a feedback survey along with a certificate that you've participated in the webinar, and that you should get that within a day. We'll make the webinar available on our GenQA YouTube channel after 1 month, and you can subscribe to our YouTube channel if you actually go to our website and info@genqa.org as well. And thank you very much for attending this webinar.